Okay, so we're going to look at protein synthesis in this screencast. When we talk about protein synthesis, we're really talking about gene expression. So I kind of use it as an overarching term to talk about all of the steps that are involved in the process of going from DNA to a functional protein, but you might also hear it just refer to the very last step where the actual protein is made. So when we talk about gene expression, which is the better term, it refers to the transfer of genetic information from DNA to RNA to protein. And that is what's called the central dogma of molecular genetics. And the central dogma is what's really at the heart of molecular biology, which is that our DNA codes for RNA, which in turn gives the information to make a functional protein. Which means that if there's a change in the DNA, there's going to be a change in the subsequent RNA, which means there's going to be a change in the sequence of the protein, which in turn is going to affect its structure and its function, because that's determined by its sequence. It's a two-step process, transcription, which is the conversion of a DNA template into an RNA strand, and then translation, which is when we take that mRNA template and we translate it into a given amino acid sequence. So the first phase, transcription, is synthesizing RNA from DNA. And transcription is the first step in the process of a gene being expressed. It involves the production of an RNA molecule from a DNA template. So DNA is double-stranded. One strand of DNA specifies the strand opposite from it through complementary base pairing rules. So if a strand of DNA, this is meant to be RNA, but if a strand of DNA has a sequence of G, T, A, C, then it's going to specify the strand that runs opposite it. So this G would base pair with a C, this T, I know it says U, but whatever, let's pretend it's T. This T would specify an A, the A would specify a T, and the C would specify a G. In the same way that DNA can code for its second half, its second strand, it can also serve as a template for a new molecule being made, that being RNA. So RNA is always single-stranded. It can complementary base pair with itself, but as a whole, it's single-stranded. And it contains A, G, and C nucleotides, same as DNA, but there is no thymine in RNA. It has been replaced by a uracil. And the sugar is slightly different. So RNA is made with ribose sugars and DNA is made of deoxyribose sugar. There's a bunch of different types of RNA. The most important ones that we're going to be dealing with are messenger RNA, or mRNA, and transfer RNA, tRNA. There is RNA found in a ribosome that's referred to as ribosomal RNA or rRNA. There's small nuclear RNA or snRNA. These are involved in splicing out of introns from the RNA, which we'll talk about in a second. So these snRNA make up protein RNA complexes called SNRPs. And then there's kind of a list of other little ones that you'd see. So the molecular events of transcription, we're going to break everything in this unit down into the phases: initiation, elongation, and termination. The process is quite similar, except there are more proteins involved in eukaryotic transcription. Eukaryotic transcription happens in the nucleus, and so it's a separate event from translation, whereas in prokaryotes, because there is no nucleus, transcription and translation can actually occur simultaneously, and we'll see a picture of that. So during the initiation phase, a transcription start site has to be recognized, and that is selected by the transcription machinery, which is a large complex of protein and DNA. Only one strand of the DNA is going to be transcribed to make that mRNA. And so that strand that serves as the basis for the mRNA being made is called the template strand. You might also hear it referred to as the sense strand. The strand that's not transcribed is called the coding strand or the antisense strand. I like the term coding strand a little bit better simply because it does exactly kind of what the name suggests. So the mRNA and the coding strand are going to have the exact same sequence, except the sequence in the mRNA is going to have U's, the sequence in the coding strand is going to have T's. So the enzyme is called RNA polymerase, and RNA polymerase attaches to a region upstream, so before a gene, called a promoter, and it's helped there by some things called transcription factors. So transcription factors are just other proteins and molecules that bind to the DNA and help the RNA polymerase find itself in the right spot. So in prokaryote, that's a sort of non-specific thing called a sigma factor, but in eukaryotes, because we've got differentiated tissues and things like that, we have very specific transcription factors. So transcription factors would make sure that a liver cell only gets, or a liver protein only gets expressed in a liver and not in your brain, and so on and so forth. So I mentioned that the area where RNA polymerase binds is called a promoter, and a promoter does what it says. It promotes transcription. So it's between 100 to 1,000 base pairs upstream, so before the gene, and it contains binding sites for different transcription factors and binding sites that are recognized by RNA polymerase in general. And promoter regions tend to be high in A's and T's because they have only 2 hydrogen bonds holding them together, as opposed to the G's and C's that form 3 hydrogen bonds. So areas that are rich in A's and T's, it's easier to unzip the DNA double helix than it is in areas that are rich in G's and C's. So this is an example of an E. coli promoter, just so that you can see the different groups here. So this is our start site of transcription where the mRNA is going to be made. It doesn't necessarily correspond to a start codon yet, but this is where transcription is going to start being transcribed. The 2 kind of key elements that you'll see in a prokaryotic, and they're similar in eukaryotes, but in a prokaryotic promoter, are this TATA box, which is a -10 element, and it's called a -10 element because it's 10 base pairs, 10 letters before the start site of transcription, and the -35 element here, which is again fairly rich in A's and T's, that's another site that's recognized by the sigma factor and the RNA polymerase. And these are what's called consensus sequences. So they tend to be found in almost every promoter, and they're pretty highly conserved. So they're quite similar in eukaryotic cells as well. Once the RNA polymerase is attracted to the appropriate start site of transcription, so once it binds to the promoter and it's kind of interacted with the promoter, the DNA double strands are going to separate, they're going to unzip, and this is now what we call an open complex. So it's open because the DNA is open as opposed to closed and base paired together. And RNA polymerase begins to add nucleotides in a 5' to 3' direction. So it starts at the 5' end and builds toward the 3' end. And that new RNA molecule is complementary to the DNA strand that it's being synthesized from. So this would be the template strand. This is the coding strand. And if we take a look here, here's my template strand, has the sequence TGCA. If you look at the coding strand, the sequence in the coding strand is ACGTC. If you look at my mRNA, the sequence is ACGUC. So the sequence of the mRNA is identical to the sequence of the coding strand, except the T's have been replaced with U's. And that makes sense because they've both been synthesized complementary to the exact same DNA template. And we can synthesize hundreds of our mRNA molecules simultaneously as long as the RNA polymerase complex has removed from the promoter region. So once the RNA polymerase moves past the promoter, another RNA polymerase can come in, it can bind to that promoter and begin transcribing, following along behind the first one. And in some cases, what you'll actually see in a prokaryotic cell is that as this mRNA is being made, a ribosome will immediately add itself to the end and begin translating it. So they're going to happen simultaneously. And you can see this here. So this is synthesis of many mRNA molecules. So transcription would be starting down here, and it would be getting longer and longer, and the little red blobs, I believe, are proteins. So I think these are the ribosomes that are attached, and the new proteins are being made coming out simultaneously. So finally, the termination, a specific sequence of nucleotides in the DNA serves as a signal to stop transcription. So the mRNA will form a hairpin loop because of the sequence, and that's going to help RNA polymerase fall off and detach from the DNA strand. The new mRNA strand is then released from the transcription assembly, and the DNA double helix will reform. So it will reform a closed complex. Now, in prokaryotes, transcription and translation can occur simultaneously. There is no nucleus, so as the mRNA is being made, the ribosome is binding to it, and it is making the protein. In eukaryotes, on the other hand, you've got an added step. So the mRNA has to leave the nucleus and go out into the cytoplasm, and it needs some protections before it can do that. So it undergoes modifications before it crosses the nuclear membrane. Once the modified mRNA enters the cytoplasm, it's free to undergo translation. But the modifications that happen to create a functional mRNA, the first two are quite easy. So we have the addition of a 5' cap, which is a modified guanine nucleotide, and that's actually recognized by the ribosome in the protein synthesis machinery. So it helps the ribosome bind to the mRNA. We add a poly-A tail. So poly-A stands for polyadenine. So we add a series of adenine A nucleotides to the end of the mRNA, and that also helps to increase its stability. So I like to think of it as though there's a whole bunch of little Pac-Man RNA degrading enzymes floating around in your cytoplasm, and the 5' cap prevents those RNA degrading enzymes from coming at it in the 5' direction. And the poly-A tail means that as those little Pac-Man RNA degrading enzymes are chewing in on it from the A side, from the 3' side, it has to chew down these hundreds of amino or of adenine nucleotides before it can get to the actual gene, so that it gives it time for the gene to be processed. And the last step is splicing out of introns. So here's my 5' cap, just shows you it's a methylguanosine, so it's a modified guanine nucleotide. And then my poly-A tail, which is 100 to 200s of A's. So this is simplified, showing you about 10. But now the removal of introns. We have these complexes called SNRPs, which is a combination of these small nuclear RNAs and proteins. And what the SNRPs do is they remove introns, which are non-coding regions in your mRNA. So these non-coding regions need to be removed, and the exons or coding regions need to be connected together. So the snRNA basically serves as a way of recognizing the recognition sequence. So it's really there because you need some nucleotides to recognize the nucleotide sequences on the introns. So what happens is it base pairs with the 5' end of the intron and a branch point, and comes together to form a spliceosome. Now, at the branch point, the mRNA is broken. The 3' end is held in place. So this kind of exon is sort of held in place by the spliceosomes. The loop in forms what's called a lariat structure. So you can see it's kind of got this like a little looping sort of jobby here. So it forms a lariat, and then the 5' end of the intron is also cut off. These two are refused, and the excised intron, the lariat, just hangs out and is degraded by those little Pac-Man RNA eating things, and the spliceosomes or SNRPs break off and go find another RNA to splice. So once the mRNA has its 5' cap, its poly-A tail, and its introns removed, it's ready to go out in the nucleus where it can meet up with a ribosome and be used to synthesize protein. Let's take a quick look at an animation.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA, called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.
Okay, so going back to translation, once we have our modified mRNA, translation can occur. And for this to occur, you need your mRNA that you made in the last step, some tRNAs, which are small RNA molecules that have kind of a 3D clover-leaf structure. We'll talk about those a little bit more in a second. Ribosomes, and some things called translation factors, which we're not really going to talk too much about. So tRNAs are stem loops, so they're single-stranded RNAs that form base pairs to create these stem loop cloverleaf structures. But the important functional regions on those tRNA are an anticodon loop and an acceptor stem. So we know that an mRNA is made up of a series of codons, of 3-letter words that each specify an amino acid. A tRNA has on it an anticodon region, which is a 3-letter word that's complementary to the mRNA codon. So the 3-letter anticodon is going to base pair to the 3-letter codon. The other end of the tRNA is an acceptor stem. So the acceptor stem of a tRNA is a single-stranded region where an amino acid is going to be attached. And when it has an amino acid attached, it's called an aminoacyl tRNA. And there are a series of aminoacyl tRNA synthetase enzymes that's their job, is to make sure that the correct amino acid is attached to correct tRNA. So there's 20 different synthetase enzymes, one for each of the 20 different amino acids. So this is kind of the way we tend to draw the cloverleaf tRNA. So you can see here we've got an anticodon region. In this case, it's got the sequence GGC. So the codon that it would recognize would be CCG. And it's got an acceptor stem up the top that will pick up the amino acid. And there are some regions that complementary base pair, and those regions help keep it in this sort of stem loop kind of weird cloverleafy structure. Now in reality, what this looks like is this. So it's not quite so pretty, and this is why we draw it like this. And ribosomes are also similar to the SNRPs in transcription. Ribosomes are made up of a combination of RNA, in this case ribosomal RNA or rRNA, and protein. And ribosomes are what does the assembly. So they connect the mRNA and the tRNA and the translation factors in order to assemble the protein. Each ribosome consists of 2 subunits, a small subunit and a large subunit, and they are different in eukaryotes and prokaryotes. So eukaryotic ribosomes are actually a little bit bigger. When we measure them, you'll hear them referred to as, I think it's the 30S and the 50S subunit in prokaryotes and the 40S and 60S subunits in eukaryotes. The S stands for a unit called Svedbergs, and that's a measure of, if you put them in a centrifuge, exactly how kind of low they'll sink in a separation gradient. So there are 3 binding sites for TNA and 1 mRNA binding site, or sorry, for tRNA, and 1 binding site for mRNA, And that allows complementary base pairing between the tRNA anticodons and the mRNA codons. So here's a ribosome with no mRNA in it. You've got a small subunit and you've got a large subunit. In reality, you never see it looking like this because it actually assembles on the mRNA. This shows you the mRNA as it's passed through a ribosome that's empty of protein at the moment. So you've got your 3 tRNA binding sites, an E site, a P site, and an A site. And again, this goes through sort of the process, which we'll talk about in a little bit more detail in a second. This illustrates what's called a polyribosome. So in many cases, you'll have multiple ribosomes attached to the same tRNA. So they'll recognize the 5' cap and begin translating, and as soon as this ribosome is far enough down the chain, another ribosome will come in, recognize the 5' cap. So you can have multiple ribosomes synthesizing proteins from the same mRNA. And we can look at the events using the same 3 kind of framework stages that we looked at for DNA replication and for transcription. So initiation, elongation, and termination. During the initiation phase, the components of translation assemble. The first thing that happens is the small ribosomal subunit will attach to the mRNA near the 5' cap and start codon. And the second thing that happens is an initiator tRNA with the anticodon UAC. So it's complementary to the AUG start codon, is going to bind to that start codon. And in this case, the start codon is in the P site. So this tRNA enters into the P site. All the other tRNAs will enter into the A site except for the start codon tRNA. In eukaryotes, it's just methionine, the amino acid that it carries. In prokaryotes, it's a derivative of methionine called N-formyl methionine. And then once that complex is formed, so you've got the small ribosomal subunit and the MET tRNA is bound in there, then the large ribosomal subunit will attach, and that forms an active ribosome complex. So here we can see, here's my first step, is the small ribosomal subunit will bind, and then my tRNA will come in, bringing the amino acid methionine, and this is sitting in what will be the P site. The large ribosomal subunit will complete. I want to go back. No. What'd I do? The large ribosomal subunit will come on, and that's going to complete the process. I just want you to pay attention to the fact that that first amino acid is methionine, so the codon is AUG, the anticodon is UAC, there's a methionine, and it's bound in the P site, not the A site. So from here on in, all new tRNAs bringing amino acids to be connected to this peptide are going to be coming into the A site. This P site is where you'll see the growing chain elongate from. So during the elongation phase of transcription, you have your 3 sites. A tRNA is going to come and it's going to bring its amino acid into the A site, and it will only fit in the A site if it has the anticodon that is complementary to the mRNA codon that's in there. So here our mRNA codon is GAC. The tRNA has the anticodon CUG, so it is complementary. So this will come in and it will bring the amino acid ASP into that growing polypeptide chain. These two will be connected, and the polypeptide chain is going to move onto this tRNA, and the ribosome will shift. Okay, so once the polypeptide is on this tRNA that's in the A site, the ribosome is going to shift over 1 unit. The tRNA that was in the P site is going to move into the E site and leave, so the E or exit site, and then leave. And now the tRNA that was in the A site has moved into the P site. Another tRNA with a complementary anticodon is going to come in, and the process will continue. So here I am going to show you another animation. This actually takes it from the beginning.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site, as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
So this one shows the assembly. I'll play this one too.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formylmethionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formylmethionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formylmethionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of 1 codon, and the transfer RNA that carried the formylmethionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Okay. So as it mentioned, when a stop codon is reached, the ribosome will grind to a halt. A releasing factor will come in and cleave the polypeptide from the last tRNA, and the ribosome will dissociate. The polypeptide is released and then folded into its 3-dimensional shape so that it can carry out its function. So there's, I think, two different ways kind of seeing this graphically. So here we've got transcription. that occurs in the nucleus. The primary mRNA transcript is made. Introns are removed. A 5' cap is added. A poly A tail is added. The mRNA moves out of the nucleus into the cytoplasm. When it's out of the nucleus and into the cytoplasm, a ribosome binds, recognizing the 5' cap. So the first thing that happens is the small subunit comes in. The next thing that happens is we've got the methionine tRNA comes in and attaches to that mRNA. And then lastly, we have the large ribosomal subunit binding. And then tRNAs recognize the codons. So tRNAs have an anticodon. They recognize the codon on the mRNA. They're brought into the ribosome. They also carry with them an amino acid, and that is used to drive protein synthesis forward. And here, this is just another way of representing the same thing. We've got the DNA sequence, and transcription produces mRNA. Translation then takes those mRNA codons and, using tRNA anticodons, synthesizes a protein. And this is the central dogma of molecular genetics. So the sequence of the DNA specifies the mRNA, which then specifies the sequence of the protein. And make sure you do take a look at the mutations because they are fair game for your test.